The lovely and talented Dr. Mark Faries is up next. And I love working with Dr. Faries because he is the epitome of the well-rounded physician. He knows radiation oncology, medical oncology, and a little bit of surgical oncology. And you can tell that he's been on all of these trials, even as an investigator on Nadina, as part of the team that brought forth herpes light, not in Europe, but in the States. He's been involved in the adjuvant, neoadjuvant, and the metastatic space. So, Mark, come on up here and educate us.
All right, thanks very much. Good to see everyone. I am the token surgeon in the meeting, so you can relax a little bit because the smart people taking a break from that and going on to the surgeons to talk a little bit. My task is to talk about TIL and sort of where that is and where that's going. I'm going to step back a little bit, not as far back as sort of the Neolithic surgical approach to all these things, but at least back a little bit further in immunotherapy and talk about how some of this works. So that is the mighty T cell, the T lymphocyte, and this is the critical component of most of these therapies that you've heard about. All these drugs and most of the immunotherapies that we have all eventually work through this. And so what I'll cover is, why did we even think that the immune system could be related to cancer immunotherapy and cancer treatment, and then proving that worked and bringing it to patients, and then where we're going from here in terms of modifications to the current TIL platforms and other cellular therapies. So we've known for a very long time that the immune system has some relationship with certain tumors, particularly melanoma. And so this is just a microscopic picture of a melanoma. The melanoma cells are the fat ones on the top, and all those little blue cells at the bottom are lymphocytes. And then they're not there by accident. So they have somehow recognized that something has gone wrong and have made their way to this tumor site. And we know that when we see this happening, even just in a primary melanoma in the skin, those patients are going to do better than patients who don't have this response. So the body is able to recognize melanoma as abnormal and put up some resistance. And so this relationship with in terms of immune treatments predates our even understanding of what the immune system was. And so William Coley was a surgeon in New York at the turn of the 20th century, the end of the 19th century. And he had noted, had been noted even well before that, that in patients who occasionally would get an infection in a tumor site, the tumor would go away, kind of as a consequence of the inflammation of that. And so he started to inject bacterial toxins into tumors and reported regression in certain cases. And they know they had given enough of this treatment when the patient had a fever of 103. That's kind of how they rated the dosing. In any event, this kind of fell by the wayside after chemotherapy and radiation came along, but it still has echoes today. And so you heard about TVEC, and this is another type of intralesional immunotherapy that we used to use, still rarely do, something called BCG. So BCG is just a TB vaccine. It's a weakened form of tuberculosis used around the world as a vaccine for TB, but we used to inject it into melanoma tumors. And so this is a woman who has hundreds of these little melanoma metastases in her leg. And you can see the little dots of blood there on the upper right. That's where tumors that have now been injected with this BCG. And she went on to develop inflammation at those sites where we injected. And we only injected probably, 10, 15% of her tumors. And then they all went away. And so what's left are just these tattoo pigment sites for this patient. And this happened not because the BCG attacked the melanoma, but because the BCG triggered this immune response, tipped the balance in her between her immune system, which was trying to fight the melanoma, and the melanoma itself in favor of the immune system. And then it was strengthened and able to go off and kill the rest of these melanoma tumors. And so that same rebalancing of the immune system is what is important with all these new therapies. So this is another one of those timelines. You've seen a number of examples of this, where for many years, decades, we had sort of nothing to offer. And then there's this explosion of new drugs, and all the ones that are in blue, and to some extent the ones that are in white, are related to the immune system and related to these lymphocytes that are the army that goes off within the body to do this. So how does this work? You can have a melanoma cell that might die, might release its antigens. Those antigens are taken up by antigen-presenting cells in the immune system that then mature, and they educate these lymphocytes, the T cells, to go off and kill more melanoma cells. And it's sort of this virtuous cycle of the immune system fighting against the tumor. It can be helped by other types of immune cells or hindered by certain regulatory components in the immune system. And so we can potentially manipulate this. And that's what some of these therapies that you may have received and certainly have heard of have to do. And so one of these ways that we've been able to manipulate this is through this interaction between the antigen presenting cell and the lymphocyte. And so these two cells get together, and the antigen presenting cell tells the lymphocyte what it's supposed to go off and attack. And there's this molecule CD28 on the lymphocyte that then is an activating signal. That cell is activated, it goes off to find the target that the antigen presenting cell has told it to go after and kill it. And then eventually it circles back around and they interact again. But this time, the T cell now has something different on its surface called CTLA4. And as opposed to the activation of the immune cell, this turns that cell off. And so one of the drugs that we have, Yervoy, it blocks that interaction. So it keeps this on, and the parking brake essentially is left off. So you've seen, the results of that and the improvements we began to see. That was the first big advance. The other type of break is the interaction between the lymphocyte and the melanoma cell itself. So there's this interaction where the lymphocyte can recognize and then know it's supposed to kill this melanoma cell. But there can be another interaction between PD-1 and PD-L1 on the surface of these cells. And that again turns that cell off. So it blunts this ability to respond. And so the Keytruda and Opdivo, these are the drugs that act at this point. But it doesn't work for everybody. So are there ways, how can we better understand this to improve the immunity, improve the ability to have the immune system fight the melanoma? So here's that interaction between the T cell and the melanoma, the dying cell. So you interact with blocking the PD-1 there. You can block CTLA4 there. But then there are all these other components to it of different cell types that are in there. Even antibodies might be involved. And so there are various ways of targeting all those steps. And as it turns out, it's complicated, lots of ins, lots of outs. And so, totally beyond me, right? Understanding all these things. It's much simpler to think about the T cell itself. Can we just get the T cells and get those to do the job? So that's the cruise missile of the immune system. And so, again, we know that the lymphocytes can infiltrate into tumors. We can take that tumor out and we can look at it under the microscope and see those lymphocytes and highlight it with special stains and such. And then we can take that tissue that we've removed and put it into conditions in the lab that favor the growth of those lymphocytes. With lymphocyte growth factors and other things that aid those cells in proliferating. They can kill the tumor in the culture wells in the lab. And then we can take those cells out and look at them and indeed demonstrate that they can kill melanoma cells in the lab, but also expand those cells into enormous numbers through a rapid expansion protocol, just special growth conditions that really amplify those cells, and then give them back to people. And you have to give them back in a very specific context where you've created space with depleting their existing lymphocytes, and then support those cells with lymphocyte growth factors afterward. But you can see that those cells that have been amplified and given back can then kill the tumors that are left in that patient. And this is not new information. So these are very old figures from papers that came from the NCI. Just exhibiting responses in these patients that can happen even with fairly extensive disease and fairly rapidly. And so it wasn't a problem anymore of knowing that this could work. It was a problem of how do you actually make this happen, because it only happens at places like the National Cancer Institute or MD Anderson, these places that you can see from space, right? They're so big that it can work there, but what about the rest of the world? And so Dr. Hamid and I, foolishly enough, created our own TIL protocol and our own GMP manufacturing type facility, clean rooms and all of that to be able to do this. And we're able to do this on a handful of occasions, but it's a pretty heavy lift, right? And this is, again, not something that's going to be able to be farmed down around the world. So how does this problem get solved? It gets solved through partnerships. So, and working with industry, actually, who has the resources to be able to potentially make this a broadly available therapy. And the initial example of this was with CAR T cells, and I'll talk a little bit more about what that is in a moment. But these are cells that are, at the moment anyway, largely addressed for liquid malignancies, lymphomas, leukemias. But there are 120 companies just looking at this treatments for this one type of hematologic malignancy. There are CAR NK cells, mentioned that very briefly, and then TIL. And there are a number of companies then that have been working to make this treatment practical to be available to the population in general. The first to the table is going to be Iovance, and we'll talk a little bit about some of their data here in a moment. So this is the trial that led to now the availability of this treatment outside of clinical trials. So this is a trial. You can see Dr. Hamid's name there on the list of contributors. And so the initial report, 66 patients, and they had already failed a number of other therapies. But we're able to get on to this treatment with the lymphocytes, 36% objective response rate, and the duration of response, very important, has not been reached in the initial reports. But, and this is one of the challenges with TIL, they still had to be lymphodepleted. They still had to have this interleukin-2, this lymphocyte growth factor, and that's where all of the toxicity from this treatment comes from, those components. So that still has to be part of it. It's a somewhat select group of patients that can have it. This is the waterfall plot from that trial. So waterfall plot is each of the bars on the plot represents the patient, and if their patient got smaller, the bar goes down. If the tumors got bigger, then the bar goes up. I think it was Tony Rebus that used to say we didn't have this in melanoma. We had geyser plots in melanoma. We didn't have waterfall plots, but now we have waterfall plots where the bars go down. And you can see the majority of the patients, there's some diminution of their tumor size. The other important thing about this, it's a once and done therapy, so they get treated and then they're not getting treated anymore. Even despite not continuing on therapy, the ones that have responses, those responses seem to last. And so that's enormously important for the therapies that we like to see. And so this led to the approval of this therapy with the FDA earlier this year. And there are a number of centers now that are online and coming online to be able to offer this therapy even outside of the confines of a clinical trial. So can we make it better? And so one of the problems is you can't get cells that recognize the tumors in all cases. So can you affect this T cell receptor, which is essentially the steering wheel that tells the cell what it's supposed to, where it's supposed to go? And there are ways of doing that to be able to generate these tumor-specific cells that don't require taking out tumors. And in these instances, these are these CAR T cells and also what are called transgenic T cell receptor cells. You can take lymphocytes just out of the peripheral blood and treat those outside of the body to make them specific for the tumor. And you can do this by changing their steering wheel, the T cell receptor, so that it's now specific for a tumor target. You can also change the cells so that they have essentially an antibody on their surface. These are the chimeric antigen receptor CAR T cells that are currently available for lymphomas and leukemias. And so this can be done to sort of redirect these cells, and you make these generic lymphocytes into tumor-specific lymphocytes and then give those back. And they have different advantages and disadvantages, different strategies. So the transgenic T cell receptors recognize targets on the immune cell, on the tumor cell in the context of what's called MHC. So that's just the normal way that cells sort of put stuff on their surface to say who they are. And there are ways that the immune system can recognize those cells as abnormal. mostly because it's putting a viral protein on there or something like that. But it's good because you can have any cell, any protein in the cell can be a target inside or outside of the cell. But you have to maintain this system of antigen presentation, which some tumor cells lose. The other option, the chimeric antigen receptor, doesn't require any of that antigen presenting machinery in the cell, but it requires the target to be on the surface of the cell. It's also not restricted to certain tissue types. So some initial data from quite a while ago now on these transgenic T cell receptors showed that you could give these cells and they could persist in the patients and that they cause some decrease in the tumor. But it was all very brief, generally speaking, in these early trials, and not a sustained reaction. It may have to do with the fact that what you're targeting is such a narrow spectrum of things that the tumors can kind of get out from under it. The CAR T cells are being used now, not just for these lymphomas and leukemias, but for solid tumors as well. Lots of different areas of research into various tumor types. With I'd say variable levels of success so far. In melanoma, or actually in these therapies in general, there is some concern about them because they can be toxic. And so you saw Dr. Sullivan with the FIRE example. These cells are alive. These therapies are alive. And so when they're given back, they can go haywire. They can sort of get out of control. And so an example where HER2, which is, about probably more from breast cancer, was the target in this patient. They got these cells specific for that, and the cells went crazy. And unfortunately, this patient had a horrible inflammatory response and died as a result of the treatment, not as the tumor. So some caution is still required for this. In melanoma, CAR Ts have been examined in a bunch of different trials. And we don't have a whole lot of results from these trials so far. I'd say the trials that are done already must not have been successful because we haven't really heard about them in terms of results. But lots of hope for some of these more recent trials. As they get done, we'll see what we learn from these. And this can be, the technology is such now that this can be taken really to an extreme. And so this is a schema that comes again from the NCI, the people that have driven so much of this research over the years, and they'll take tumors and they'll do whole genome sequencing on them and identify all the mutations and then screen for cells that are responsive to those specific mutations, or they'll potentially generate T cell receptors that are responsive to those mutations and give those back to the patients. And they have certainly seen with this really extensive effort clinical responses. Unfortunately, even now, they're not super common with a fairly low rate of partial responses so far anyway. But we'll see. Everyone always said that TIL was the therapy of the future and always would be. And that's come to fruition. So we'll see if the NCI brings something else as time goes on. There are lots of ways that the TIL therapy, the current standard TIL therapy, might be modified to make it either more effective or more tolerable. You might change the timing of it. One of the issues is there's a delay between when the tissue is taken out and when the patient actually receives the therapy, and that delay can be too long for some patients. And so can you give it earlier in the neoadjuvant or adjuvant setting potentially? Can you reduce this lymphodepletion, which has some side effects? Can you make that more tolerable? Can you change the cells you pick to grow? And there are a few efforts to do that. And can you combine it with some of these other therapies that we have? Can we change the fecal microbiota to improve this? So you can see there are many ideas about how to modify these cells or modify the conditions that they're given in to make things better, even than the good results that we've seen so far. So that's the T lymphocytes. There are other immune cells that may also be useful for adoptive transfer, that is, taking the cells and giving them to patients. NK cells, you heard a little bit about from Dr. Hamid. NK stands for natural killer. So that's much better than herpes light, right, as a marketing tool. But these cells have the ability to recognize certain innate things that may be present in cancer cells. They're not antigen specific, so they're not against a specific tumor antigen, but they can sometimes recognize cells based on changes that happen in cancer cells. So, for example, some cancer cells, to escape the immune system, lose this antigen presentation mechanism that you saw before from the cartoon earlier, that's a trigger actually for NK cells to identify them and kill them. And so you may be able to chase these tumor cells, force them down one sort of resistance pathway, and then hit them with NK cells, for example, after that. And so there are some data actually very early on. There were what were called LAC cells, lymphokine activated killer cells, treated at the NCI before they got the TIL stuff working. And about 10% of those cells were NK cells. They saw some responsiveness, but not really impressive clinical results. But some hints that there may be some clinical effectiveness of this. You can modify those NK cells, and then there's a separate type of cell called an NKT cell that also can potentially recognize sort of innate changes in the way cancer cells behave that might make them amenable to other forms of immune recognition and destruction. So the standard TIL now seems ready for prime time. It's going to be available much more broadly to patients than it has been in the past, and so that'll be a great thing. We need to figure out where that fits into the overall treatment paradigm. Right now, it is only for patients who have failed other therapies, but maybe that needs to shift around a little bit. And then there are potentially ways to modify these therapies to make them more effective over time. CAR-T, I would say, for solid tumor, still many challenges to be overcome for that, but certainly an active area of investigation. And then NK and NKT cells, I think, interesting. Jury's still out for now, but a lot of interesting trials that are ongoing. And so we'll see how those turn out. So, we get to be the spokesmodels for these things, right, for all these therapies. But it takes a huge number of people, and many of you have met these people. And whether it's the person that draws the blood, starts the IV, puts you through the scanner, checks off your con meds when you come in for your clinical trial visits, it requires this huge effort of all these people. And so this is who deserves a lot of credit for all these advances that you've seen. And it's an honor for us to work with these folks as well. So thanks for being here.